Welcome to Soil Smart, Soil Wise webinars. Today we're going to be learning from Nick Tomasini of SoilWright, W-R-I-G-H-T. He's in Oregon and he has a company that is creating compost. So he's going to share a little bit about what he's learned about compost. He's the founder of an organization called Humankind in Oregon, that's an LLC. And he focuses on compost production and soil and crop consulting to support healthier farms through biological regeneration. He has a BS in agriculture from the Oregon State University and training from Dr. Elaine Ingham from the Foodweb School, which I highly recommend and we're offering scholarships to. Nick has spent over 2 decades working across organic farming, soil fertility, research, and organic inspection. As a certified soil Foodweb consultant and agronomist, he has collaborated with farmers and compost producers to better understand and apply soil biology in practical ways. Now, I have to give you a heads up. This is a very high-level understanding of compost, and yet what he has to say is really quite miraculous. So I hope you'll stay to the end. We had a lot of discussion, but there is a lot to learn. This is part of our webinar series, and I urge you to look at our website and under events to see the free webinars that are happening currently. So I'm Catherine Alexander. I'm the Chief Strategist for Soil Smart, Soil Wise. Let's take it away. Yeah.
That's okay. We have days. So this was about, let's say, an 85-day process, a little longer than what we're achieving now. And then on the Y vertical axis, we have degrees Fahrenheit. So let's just kind of run through these various stages: mesophilic, thermophilic, mesophilic 2, and then maturation, and kind of the way we think about it and what's going on there. So in mesophilic 1, this is, we've just constructed a windrow. We have very diverse food resources. The way we do our recipes, which we'll talk a little more about in the next couple of slides, is we start with a pretty high carbon to nitrogen ratio relative to maybe what some other composting operations are starting at. So in the 40-to-1 range, sometimes all the way up to 48-to-1. And we've got a lot of microbial activity that builds very quickly here in the mesophilic 1 stage. So you can see over the course of 3-4 days, we're already up above 120 degrees and into our thermophilic stage, which was a little longer for this particular windrow. It's the one I had prepared, and I think it's a unique one. We had some issues in the middle. They didn't persist for too long, but we'll be able to talk about some of that. So this thermophilic stage, again, 113 to 176 degrees Fahrenheit is the way science actually defines thermophilic. We like to be somewhere in the neighborhood of 125 to, maybe just over 150. Sometimes we'll touch 160, but we kind of try to stay away from that with our higher carbon to nitrogen ratio. So again, an aerobic process, a lot of activity going on here, mostly bacterial, but we have actinobacteria, archaea, fungi also kind of in the mix here. But the rapid activity and reproduction of these organisms is what is actually creating this heat. And just to kind of put that in perspective, these bacteria can reproduce every 20 minutes or so. And another way to think about that is they can essentially double their population every 20 minutes under the right conditions. So just a ton of activity, a ton of energy conversion and expenditure here, a lot of enthalpy and quite a bit of entropy too. There's a ton of oxygen that's being used, so the oxygen demand is really high. And as you see, we get to about day 7, we're up at about, 137 degrees or something. We really only like it to be up in that 130 range for 3-4 days max before we turn it. And the reason we're doing this is because we need to reintroduce oxygen. There is so much activity going on, and we want to keep this process as aerobic as possible. There is a ton of CO2 being respired as well, which is heavy. And so that can sit in the windrow, and you can get suffocation of these aerobic thermophiles, and essentially you can go anaerobic very quickly. So we really try to leverage this thermophilic stage for rapid decomposition, and it's really the simple foods in our recipe that are used up first in this stage. So we're talking about protein, nitrogen from manures, from legumes, things like that, cellulose, simple sugars, those kinds of things. These thermophiles are very opportunistic, very fast growing, and they're breaking this stuff down at just a really rapid clip. We can.
Nick, can I ask a couple of questions just to make sure everybody's on the same page?
Yes.
Um, so when you talk 40-1, you're talking carbon to nitrogen, right?
I am talking carbon to nitrogen ratio, yes.
Okay, just to make sure that people understand that. And oh geez, there was another thing that hit my mind.
We're going to revisit a lot of this stuff too, but yes, please, if anything is unclear at this point.
I'm just because you are really starting at a very high level around compost. A lot of people are much simpler, and so I want to make sure that they come along on this ride.
For sure. For sure. So I'm going to switch away from this graph really quick and just kind of look at some thermophiles here for a second. So these are the microorganisms we're talking about that are doing this rapid decomposition. So again, archaea, bacteria, actinobacteria, and fungi. And they're excreting a lot of extracellular acids and enzymes and various polymeric compounds that allow them to basically degrade and digest this material. And that's how they're able to, that's where they're getting their energy for reproduction, etc. And again, if it wasn't clear from before, the oxygen demand is so high during this that we're really, and the reason we're turning so frequently, as you'll see through this stage, is that we need to reintroduce that oxygen and allow that CO2 to dissipate a little bit. So we keep this an aerobic process because, as you'll see, we're really working on a microbial succession here. So we want to have beneficial fungi, protozoa, and nematodes, which we'll take a look at as well in our finished product. And in order for that to happen, we need to leverage this thermophilic stage so we get a lot of decomposition, a lot of nutrient release, etc. But we don't go anaerobic so as to hinder the appearance and the proliferation of this beneficial fungi, these protists and these nematodes that we'll talk about sort of toward the back end of thermophilic composting. Go ahead, Catherine.
Okay, another question, because you mentioned hedgerows. So a lot of times when people do compost, they do piles. So
Yes.
would you take a minute to talk about the difference between piles and hedgerows, and maybe occasionally, if that's relevant, how that might differ in what you're talking about?
Yeah, so in this first picture, let's just go back to this. This is a windrow turner, and a windrow is essentially like a long pile that's peaked. So it's a long peaked pile. And in this picture, we're turning a windrow that's pretty early on after construction, and we're actually injecting it with compost extract, and we'll get to that later on. But this windrow turner essentially turns the compost, and we're able to add moisture or put whatever else we want into the compost with this turner. So this is how we make sure that all of these organic residues, crop residues, manures, etc., make it through the hot center, because the hot center of a windrow or a pile is really where a lot of this thermophilic activity and decomposition is happening. So for us, we're not, I wouldn't say we're large scale, we're mid scale. We use windrows, these long peaked piles. But I have been making compost for the last 10, 15 years, and the majority of that has been in a pile. And, different systems. I've done a lot of vermicomposting as well, which is not hot composting, although sometimes I'll do hot composting first, and then I will feed that compost to the worms as my vermicomposting process. But yeah, I mean, the way I really learned to compost was in a pile. And so I'm kind of giving you the scaled up version of this, but we're using a lot of the same principles in our composting now as I did early on when I was learning how to compost. And really, like the pivotal moment for me was learning how to compost from Dr. Elaine Ingham. And I've taken what I learned from her, and it was a lot of failure and just learning from those failures in the beginning, and built upon that over time with various systems, and now scaling this system here. So yeah, there's a lot of different ways to do this. There are larger scale ways to do this. There's anaerobic digestion where um, you're you're not really using a hot thermophilic process. It's an anaerobic process, so no cellular respiration, and the byproduct is methane, which is being captured and utilized as an energy source. So, these very large scale livestock operations, probably a lot of CAFO operations too, they have all this manure. Yes, it would be best if it was out on pasture and in the grasslands where grazing should be happening. But they build this stuff up, and it needs to be composted. And that's one way they kind of convert it to energy. But they also do thermophilic composting and probably a little worm composting as well. But again, I'm giving you sort of our take on thermophilic composting, and perhaps it is high level. So please stop me if it gets if I'm going over people's heads here. But I do assume that some of the people here are familiar with composting and some of these methods as well. So again, ask questions as we go here.
And Charles has a question.
Go ahead.
Yeah, so is that anaerobic process, so that be a biodigester type of process where you're capturing the methane? Is that, am I thinking the same thing?
Yes. Yes, exactly. And I think, while it's been around for a long time, it's becoming more popular now as we look, to alternative energy sources, as we see sort of the detriment of methane, which is more disruptive to the atmosphere than CO2 even. So they're trying to utilize this, capture it for energy. I'm not sure that, I haven't really studied closely the carbon footprint on that whole process. I would assume that it's better than just kind of letting it sit in a lagoon or a pile for extended periods of time until all the methane is gone. But I don't actually know the answer to that. I haven't studied it enough.
I would actually wonder too about the micro balance in the final product.
Yeah. Well, same. And I'll tell you that it's probably not going to be the same outcomes that we're able to achieve with this type of a method, but there may be something you could do on the back end with that material. I do know that aerobes can grow somewhat effectively on products that have been fermented wood chips, different things like that. So I know that as things come back to aerobic, even if they've gone through an anaerobic process, they can still be viable as a food source, maybe a soil amendment too, but as a food source for aerobes. And you would see aerobes come back into that, but I don't think it would be a wide diversity of microorganisms. So definitely not like a fully functional soil food web. But yeah, probably some aerobic bacteria would come into that scenario. If you fed it to worms, if it was aerobic for long enough afterwards, and they didn't scurry out of there, which they will do sometimes if something is too anaerobic and they don't like it, then perhaps you could improve it through vermiculture. But I'm not sure the large scale operations who are doing this type of anaerobic digestion are really doing that sort of thing. I think it's more energy production. They're using a waste stream to make energy essentially, and probably profits. So I'm not sure soil health is a big part of that picture. I think it's a diversion of methane and profit, energy, etc.
And I think that's really important to emphasize actually, because what we're really promoting here is compost for soil health. And I think often even beginning composters see it more as a way to use yard waste, not so much as an actual nutrient mechanism. So they're not thinking about compost as having nutrition or paying attention to that. That's for me one of the real benefits of the Soil Food Web School's approach. is that you really begin to think about the richness of life as being necessary and paying attention to your compost as having that, because it doesn't always, even under normal circumstances. It may only be partially sufficient.
That's my
Charles?
experience. Yeah, go ahead.
Charles?
Yeah, no, I've been I've sort of seen some small scale operations in South America using the biodigesters, and they were fully using it for fuel, and there was sort of like there was that secondary product, and it was also very hard to keep the balance correct in there. Like these people were having trouble to actually do it. I mean, that's just my comment there.
Yeah, I'm sure, just like these considerations, this talk is really about, how to generate this kind of compost, these microbial outcomes for soil health, but like all of the considerations as we go through this and some of the pitfalls as well, we'll get into if we have time for that. But so these are the thermophiles that are sort of carrying out the first leg or the majority of this thermophilic composting process. And so top left, you've got cocci bacteria, that's thermos. And so we do some DNA testing as well. Every compost I've seen has contained some thermos genus bacteria thermophiles in it. So they're very ubiquitous. And then the one on the right is actually Bacillus subtilis, which is a facultative thermophile. So basically, it can live under these live and function under these hot thermophilic conditions, but also in ambient conditions as well. And with Bacillus subtilis, it's very common as a soil microbial inoculant, as a biocontrol of some fungal pathogens. And even the other day, I saw that it was in the kombucha. They had added Bacillus subtilis to the kombucha as a gut probiotic as well. So that's what the specimen is there on the top right. But getting into some of these,
Francesco, did you have a question?
I don't think.
Let me see if I can unmute you here. You should be able to unmute, Francesco.
I don't think he had a question.
I have a question.
Okay. Okay.
He was-
All right. Sorry.
he was
Sorry to
just
interrupt.
Stretching a little. So conventionally, oftentimes with these thermophilic composting operations, they're looking at this thermophilic phase not just as a way to do rapid decomposition, but to also aid in pathogen and seed reduction in the compost. So if you're putting it in your garden, if you're putting it on your farm, you're not getting any weed seeds there. You're not actually transplanting pathogens into your soil. And there are different types of thermophiles as well. There's simple, extreme, and then there's hyper. And these like thermal vents in the ocean, et cetera, that's where you're going to find these species like hyperthermophiles, but we don't really have those in our compost. Again, I've seen compost get up into the mid 170s before. And if it goes anaerobic when you're up hot like that into the 180s and starts producing alcohols and various gases, you can get spontaneous combustion. I've even seen large piles of wood chips spontaneously combust. So we're trying to avoid that, but really we're trying to keep it aerobic and not get too hot so that we can still have this microbial succession, this diverse microbial outcome with these eukaryotes we'll kind of talk about in a minute in our finished product. So we're leveraging this thermophilic stage to get hot, do rapid decomposition, make nutrients available for other microbes through the next stages of this process. So going back to our graph here, where you see these valleys in our curve, our sort of jagged curve, that's where we turn and the temperature goes back down, right? So you can see, within the first like 7 days we turn, and by like day 12, we're at 150 degrees again. So it shot back up really quick. That's how much and that is the activity and reproduction of these thermophiles as they break down and decompose this material in the compost. So we're up there pretty high, about as high as we really like to see it go at this point. And we're only going to let it sit there for like a day or two, maybe 3 days in some cases. early on, especially. So we turn again, and you can see that by day 15 or so, we're back down below 130. And then the slope of our line changes quite a bit. And it takes us a while to get back up above 131, which is kind of a threshold for us. And within the organic regulations too, they want compost with manure in it to be up above 131 for a certain period of time, or they simply will call it manure. It won't be classified as compost, and there are restrictions around manure use. That was an aside, hopefully not too much of one. But anyway, the slope of our line changes here. And what ended up happening was we were dry. There's plenty of food resource left in here. We just did not have the moisture. And so it took us a little while to climb back up. By about looks like day 27 or so, we top watered. So we actually added some moisture. And you can see that we got a nice spike in temperature there, and we turned after another 3 days. And then after that turn, quickly back up above 150 again. So that was our signal that we just needed, these organisms needed more moisture to do that work. There's plenty of food there, plenty of microbes, plenty of oxygen. We just didn't have the moisture. So we shot back up as a result of all of these food resources, and we turned again after a few days, and then the pile started to mellow out a bit temperature wise. And so we get another, looks like 2 peaks and turns in the 130s through our thermophilic stage. And then we start to decline. And we would like to see a 20 to 30 day thermophilic window. That's kind of our goal for our windrows. But this one, with that sort of hiccup in the middle there, ended up being more like 40 days, and which is totally fine. This was still a really good microbial outcome in this compost. So now we get into mesophilic 2 as we drop down below about 113. And so the activity, the heat, and the simple foods have all decreased at this point in time. So without the food resource, the activity is not as high. They're not producing as much heat. The competitive exclusion that these fast-growing thermophiles were enjoying before is dissipating. They're just much more aggressive at getting after those simple foods. So now what we see is more diversity come back into our compost. So more diversity in the bacterial species, so they don't have to be thermophiles anymore once we're dropping below 113 or so. And then we also have some of our keystone predators showing up, so protozoa and nematodes. And so if you're familiar with like keystone species or keystone predator theory within ecology, introducing predators to a system if they've been gone, and maybe certain species are enjoying some competitive exclusion, can really control that situation and increase diversity. And that's exactly what we see as these micro predators come in. And when I say protists and nematodes, let's just take a little look at them here. So protozoa, ciliates, amoeba, and flagellates. Sometimes we'll see some paramecium and some rotifers in our compost, but not as often. We really focus on these 3. They essentially feed, so these are the predators. They feed on bacteria and fungi and organic matter in some cases, and they release nutrition, so nitrogen in the form of ammonium, which is a plant available form when this is happening in the soil. These predators are not only improving diversity, but they are aiding greatly to nutrient cycling in soil. So if the bacteria and our compost and our soil are holding the bulk of our nutrition, then we need these predators to come along and release it more efficiently in that rhizosphere, and the plants can access it at that point. And we talk about ammonium here as the form of nitrogen because in science everything is kind of nitrogen centric when it comes to nutrient cycling, but we're talking about the carbon, the nitrogen, the phosphorus, the sulfur, etc. And so they're very important for nutrient cycling and plant growth and all the biogeochemical cycles on this planet. If the bacteria are absorbing and immobilizing, the protozoa are coming along and they're mobilizing or mineralizing that nutrition. And then it goes back into this ecosystem for plant uptake. Other microbes can uptake it and utilize it for their growth, etc. Now we've got a functional ecosystem going on here. And so let's just look at our nematodes too, since they tend to show up. Nematodes are are roundworms, microscopic roundworms, and they can be bacteria. They can eat bacteria, fungi. They can be omnivores where they eat bacteria and fungi and even plant roots in some cases. We have predatory nematodes that will eat other nematodes, plant parasitic nematodes. And if we look at the bottom right picture, you can see that one. If you can see it well enough, it has like a spear inside of its mouth or a stylet, and it uses that to actually puncture plant roots. So we don't really see these in our compost, firstly because we don't have plants in our compost and they need a host to do their thing. But we do see them in soil, and I've had to deal with them on the soil side quite a lot. And I just had a decent picture of one, so I thought I would throw it in here. But the two on the left, those are bacterial feeders. So very similar to the protozoa that we were just talking about. They are eating bacteria, and releasing plant available nutrition or nutrition that's available to other microbes within that microbial food web. And the one on the top right is tough to see, but it's a predatory nematode, and they essentially prey on other nematodes. So any kind of nematode really that they can fit into their mouth is what they'll go after. But yeah, so again, these are some of the predators that start to show up during this mesophilic 2 stage. And I should mention, these are 4 types of amoeba in these photos here in this slide. The flagellates are very small, and the ciliates I had video of, but I didn't have any good stills. So I ended up just putting some of these specimens of testate amoeba in here. And they are, again, they're single-celled, but they're eukaryotic, so they do have a nucleus. They're predators, and they build these shells out of siliceous materials within the soil, and they live inside them. So it's kind of protection and just a harbor from adverse conditions. And the reason a lot of these protozoa will make it through that thermophilic stage, because they're non-thermophiles, is that they can produce cysts. So they can go dormant in a cyst form and resist these very hot temperatures and, this biochemical profile being put out by the thermophiles. And then once conditions are right, they can, and their food source is present, they will come out of dormancy and become active again. So at this point, there's been a lot of disturbance through that thermophilic stage, but we needed to leverage that and do that disturbance, that turning, so that we could keep it as aerobic as possible. But now, as we get into mesophilic 2, we really want to limit that disturbance, because especially on the fungal side. So you can see we turned it 2 more times through the mesophilic stage, and we actually implemented a cool down protocol. We can talk a little bit more about that later on, but we wanted to cool it down a little more based on the good level of decomposition we were seeing, the presence of a lot of these microbes we've just been talking about showing up at that point in time, and we just didn't want to disturb it much more. We were seeing the fungal biomass and fungal activity increase during this mesophilic 2, and they just do not do well with a high degree of disturbance. So this is really the time for these predators and the fungi to proliferate, and we want to minimize that disturbance.
Nick, how do you know that these things are in the soil? I mean,
very good question. Yeah, very good question. So the main method we use for verification, for quantification of these organisms, and to track soil health on a microbiological level is microscopy. So direct microscopy primarily. We will do a little bit of root staining and some epifluorescence microscopy when we look at mycorrhizal colonization of roots. But we can't actually produce mycorrhizal fungi in our compost because we don't have that plant host in here. So that is one of the players. If we were to say we want this to be a completely functional microbial food web, we don't have that mycorrhizal component. So when we go out to the field, that is one inoculant that we will add. And we've talked about maybe offering a product where we put it in after curing and maturation, so it's there in the product. But we'll see, maybe. So as we get into maturation, you can see, we're down below 100 degrees now. And typically we want this to at least be a 3-4 week process with no disturbance here. Most of the cellulose these simple carbs and sugars, the protein, those have been consumed and turned into biomass, and a lot of that biomass is turned over and decreased at this point in time. A lot of these predators are further decreasing that explosion of bacterial biomass that happened during that thermophilic and mesophilic 2 stage. And what we're left with, because of the way we do our recipes, is a lot of woody material. So very complex carbonaceous type material that's high in lignocellulose and lignin. And bacteria don't really make the enzyme profile to break this down effectively. They will try, and they will have a little bit of success, but the nitrogen just isn't there for them when they try to do this. Bacteria are the highest carbon to nitrogen life form on this planet, somewhere between a 5-to-1 and an 8-to-1. So they need a ton of nitrogen to do their thing. And, this lignin is maybe 100, 150, 200-to-1 carbon to nitrogen. So it's the only food that's left. They try to break it down, but they can't really extract much energy from it. They're just not evolved to do that. So this is the time we see fungal proliferation occur, as they are the specialists that can break down these types of materials in our forests, et cetera. Maybe you've heard people say that we'd be, swimming in lignin and woody material debris from trees if it weren't for fungi breaking it down in our forests. So they make a very specific concoction of organic acids and enzymes, manganese peroxidases, et cetera, that are specialized at breaking down this lignin. So we've sort of set the stage. We had decomposition, very rapid, but we kept it under control, so it stayed aerobic. The food resources were depleted. We got that good level of decomposition, so we created a nice substrate. Our predators started to show up. Our diversity started to increase. Now our food sources have depleted and have become kind of narrow again. And fungi are slow growers who are specialized at breaking down what we have left. And we set up our windrows exactly for this reason, so that we get this fungal proliferation on the back end through this mesophilic 2, but especially in these, through the maturation stage. And so let's just take a look. So here are some photos of fungal hyphae. And these are essentially like the individual, these are, 4 different species that we see in compost. But these are like the individual strands that make up a mycelial network. And these are saprotrophic fungi. So they're not mycorrhizal, they're more like decomposer fungi. But they do a lot for soil, just as they're doing in our compost here. So they decompose the more complex materials in soil. Bacteria are capable of this too, but they're very effective at biomineralization, both induced and controlled. So they can precipitate and nucleate minerals, like put ions together, make minerals. The field of micromediation is starting to take off, and maybe we can talk about that a little bit more when we talk about biochar. But they're very good at solubilizing things like phosphorus and humification. So we talked about wanting stable humic compounds in our finished compost. And this is what we want in our soil too. We want stable carbon in our soil. So fungi are very, within their mycelial networks within soil, there is a lot of carbon stored there, especially when we start looking at the ectomycorrhizal necromass within forests. These are not included in any climate models, but there is a massive amount of carbon stored within that biomass and that necromass within forest soils. But coming back to humification, we've got soil organic matter or organic matter as a pretty broad pool that we hear used in a lot of contexts with soil health, climate change, just farming in general. But there are a lot of pools within that, some of which are labile or more available. Like there's, water extractable organic carbon is one thing they'll call it. And that's readily available for microbial usage and plant usage as well. And then we get into the other end of the spectrum with humus and stable humic substances. And these are very complex structures. They are phenolic aromatic rings that are really tough to break down. There's really nothing that breaks this down effectively. Long branched chains of carbon and rings that the fungi are able to polymerize. So it's essentially the polymerization of organic materials. And then they're doing biomineralization, and they're also wrapping this humus in minerals. And so it's mineral associated or mineral included or protected humus. So very stable forms of humus in our soils. And fungi and some bacteria too are specialists at this humification. So this is very important for soil carbon and soil health. And we're missing this in our compost. Most of the compost that's out there and commercially available, yes, and in our agricultural soils as well, from my firsthand experience. So we put a lot of emphasis on this group, on these predators, and this saprotrophic fungi. This is really what we're aiming for in our finished product. And just building intuition. It's hard to say what you're going to. It just takes repetition and practice. You don't really know what you're going to have as a microbial outcome or a fungal biomass until you get there. And we've got some tricks where if we're not hitting our targets, we can do some things to improve fungal biomass, et cetera. And we're learning more about that all the time. But we certainly have more to learn in that regard. So the other thing they're doing in soil is just they're creating macroaggregates. So soil structure, which allows for water infiltration, better moisture holding capacity, better gas exchange. So CO2 going out, oxygen coming in. And that is just I think it's an overlooked component of soil health. It's just the structure of that soil. Yes, roots are doing this too. The organic matter is aiding in this process through these organisms, but these strands, these mycelial networks and these strands of hyphae are like scaffolding in the soil. They're making these aggregates, they're creating structure in soil, and that just has so many soil health benefits and implications. So let's see here. I think we're pretty much finished up with this example here. I don't know if anyone has any questions, but I just thought it might be nice to kind of run through these various stages in a windrow and talk about what's going on, what we're thinking about, what we're trying to optimize for, and then meeting some of these microbes or putting a face to some of these names.
Charles has a question.
Go ahead, Charles.
Yeah, a really basic question. So are you using temperature as your main indicator of when to turn, or is it more of a time-based thing? And I assume that would change different times of year, different locations.
Yes.
So is it really?
Yeah.
Yeah.
Yeah, so we'll talk about seasonality a little bit too, but you're right. It's really temperature and time. It's both of these things. So again, really quickly, like some guidelines for this are once we're in that 100 and let's call it 125 to 140 range, we're really not going to let it sit there for longer than 3 to 5 days before we turn. And we can be more specific than that. Sometimes it's only 72 hours, but what we're finding with the way we build our recipes and having some better structure in our windrows, we can go a little bit longer than some of these windows that, for example, the Soil Foodweb school teaches, but we don't stray too far from it. Once we get up above 145 and the 150s, we're looking at more like 2 to 3 days there. We don't want to stay there for too much longer than that. And then once we get 160 plus, we're looking at a day or two. If we're hitting 170, we're looking at turning immediately, basically. So it's temperature and time is the way we're managing. And there are other ways to look at it. You can monitor oxygen, you can monitor CO2. There are management systems out there that do just that. And I've consulted with some operations that focus really closely on that CO2 output as an indicator of when to turn. And where I've seen some, with that particular client, I've seen where they can produce pretty good protozoa, but they have a hard time getting that fungal biomass at the end. And there could be other reasons for that too, but they're also injecting oxygen into their windrows and turning. So they feel like they can sit for a little bit longer. They manage that CO2, but for whatever reason, and I think it's because they're just not turning frequently enough, CO2 is building up too much. Maybe they just need to move their targets a little bit. They're not getting that high fungal biomass outcome in their finished product.
Nick, are there tips that you might offer to people who maybe don't have such sophisticated ways of doing compost and are kind of like doing it at home?
Yes.
To think
Yes.
about how to get that fungi in there, because that is so key.
Yes. Just a little pit stop on this slide. I borrowed this one from Dr. Ingham. It's been used over and over and over, and maybe some of you have seen this before, but it's basically a look at plant succession in nature and what happens with bacteria and fungi. Just a little snippet here, but on the far left side, you've basically got a rocky, sandy soil, very little plant material, and just bacteria. And as you move into a little bit more vegetation, as we move to the right, you get some fungi showing up. As you move into sort of this middle ground here, where you've got these low growing shrubs, you in nature you get a pretty balanced, fungi to bacteria ratio. And so once we move into the forest, more fungi, and once we get into later stage forests or even old growth forests, we can be 100 or 1,000 to 1 fungi to bacteria. So just to put that into perspective, early on in succession, we have just bacteria. We get more and more fungi as we move towards an old growth forest. And as you know, a lot more carbon storage in those old growth ecosystems as well, thanks in part to these fungal partners here. But what else did I want to say about this? I would say with our compost, we're somewhere in between like that third bay and that fourth bay. So that's about where we get in terms of our fungal biomass. It's usually a pretty balanced fungal to bacteria ratio in our compost. Sometimes we can get all the way up to a 1-to-1 or a little over a 1-to-1, but it's pretty tough. I think if we went slower and just used wood chips, took longer time to do this, we could have more fungal biomass. But I don't know if that thermophilic stage that would be robust enough with that to really get the level of decomposition most people expect from a compost. But there are ways to do it. I don't think anybody is doing that commercially. We're trying to push that fungal component as much as we can. And we're pretty happy, frankly, with where we're at right now. But I thought this might be a unique slide to show. So getting into just really quickly on our recipes and our feedstock proportions. So the upshot is it's really lean in nitrogen and carbs, and it's high in carbon and lignin. So again, that nitrogen and that carbohydrate, so those proteins, those aminos, those sugars over there on that side of things, that is really what is fueling these fast growing thermophiles and bacteria within that hot state, the hot thermophilic stage of our compost. So we don't want too much of that in there. And with most operations, the proportion is much higher of these types of things. So manure, green grasses, leaves, food waste, those kinds of things, they would sit in that high nitrogen category, feedstock category, and that green category. And that usually is the bulk of the feedstock for compost. And for us, it's different. We are focused on this carbon and lignin component to temper that thermophilic stage, stay anaerobic, and to have that food source for that fungal proliferation through maturation. So that's one way we think about it. But the other thing is, we use a lot of wood chips. We use straw. You could use wheat chaff, anything that is high carbon. We'll come to the categories again here in a second, but structurally speaking, we want some biofines, some fine material, like manure is a very fine biological material, where our wood chips are coarse. They're big, they're chunky. Sometimes we even have ramial chunks in there that are like this long, and it acts like scaffolding or aeration within our windrows. So it makes that whole process of keeping it aerobic a little bit easier, while at the same time having that food source there for the fungi on the back end. So yes, supports aeration and fungal colonization. And in a nutshell, we set up our recipes to leverage that thermophilic decomposition followed by the eukaryotic succession. And we really, right now we're doing things by volume. We're going to get to a point here shortly where we're tracking the weight of our feedstocks, but we look at 3 distinct feedstock categories. So pretty basic here. High nitrogen feedstocks, which would be like manure, could be grains, could be legumes, anything that's about 15, right in that 10 to 15 to 1 carbon to nitrogen, we would consider a high nitrogen. And we only add about 10% by volume to our recipe of that type of feedstock. So very low. I mean, this could be 50%, 60%. In some cases, if you have a lot of manure, I mean, maybe it's 70, 80% of your quote unquote compost, right? But without that diversity in feedstock, that food, without that high carbon component, you're going to have a very bacterial dominant type of compost, in my experience. And then we've got a green category kind of in the middle, and I would say that's sort of like a 30 to 1 to maybe like a 45 to 1 carbon to nitrogen ratio. So these are like green grasses and leaves, and food waste often fits into this category, depending on what it is. And so we're usually about 35% on the green feedstock category. And both of those, those first two, we try to, both the green and the high nitrogen are fuel for that thermophilic heat and that activity. So we tend to lean more into the green feedstock category than that high nitrogen category to do, to get the outcomes we're after. And then the high carbon, and I will say, just to kind of go back to Charles' question, there's a big seasonality component and age component too to these first 2 categories. Well, all these categories really, but aged manure might be much higher in a much higher carbon to nitrogen ratio. A lot of that nitrogen maybe is volatilized. Those proteins aren't really there anymore. It's a lot of just like the fiber from that animal's diet that's left in that. And so you've got to factor that in when you're working out these proportions. If you think that it's slipped in, I mean, manure at some point is going to be more in this high carbon feedstock. If it sits for several years just out in certain environments, there's not going to be much left in there but that fiber. And the same goes with this green category with like grasses. A spring grass is very high in sugar and cellulose and even nitrogen, because they store a lot of nitrogen in their roots over the winter, and then when that spring flush happens and that grass is really, really lush, that's almost a high nitrogen feedstock at that point. But as we get into August, depending on when your bioregion receives its rainfall, for us, we don't get much rain in August, so the grass turns pretty brown actually. And then maybe we decide to, if that's the type of feedstock we're using or we have access to at that time, maybe we decide to go up a little bit in our proportion of green material, because it's a little higher C to N. Some of that sugar, some of that cellulose has dissipated, has volatilized. It's not there anymore. So we might use a little bit more, and then account for maybe 25% of that grass actually being in the high carbon feedstock category, which we'll talk about next. But so high carbon is like the wood chips, straw, not hay. Hay would be green, straw would be high carbon, typically. There's no grain. Well, there shouldn't be grain on your straw. Sometimes the straw bales I buy, there is still grain on it. But chaff from wheat, so the shell, all these sort of like, let's say like 75 to 1, all the way up to 250 to 1 carbon to nitrogen ratio. I think the heart of a redwood tree, when it's dried out, could be like a 500 to 1 carbon to nitrogen ratio. So we're not using that type of a feedstock. That should stay in the heart of the redwood tree, frankly. But we are using some, some that are up in the 100 and 200 to 1 range there. But that can be 50 to 60% of our feedstock and of our recipe for any given windrow. So as you can see, we mean that when we say we use a lot of carbon, a lot of complex carbon, a lot of lignin in our recipes. It's like 50, 60%, 55 to 60 is typical for us. So anyway, that's that's how we manage those stages that we talked about before and get these microbial outcomes that we're after. These are just some of the ways we think about our recipe development formulation. And then here's just some hurdles, and maybe this will help some people if they're doing composting at home, or maybe it'll push them to dive into composting a little bit more as they learn here. But with our recipe, what we find is too much in and cellulose. With typical commercial operations, they're just adding, and even home composters, they're adding too much nitrogen and too much green or too much cellulose. And what they need is more complex carbon and more lignin in there. And for windrow structure as well, as we discussed before, that gas exchange component. The feedstock condition, we just talked about that. So I guess we've covered that already, but that seasonality component and the variability in seed in that can occur throughout the year. And, you know, if it's hay from a high desert region versus like a valley that's receiving 40 or 50 inches of rainfall, there's going to be a difference in carbon to nitrogen there. Usually, unless it's really low quality from the high desert, we can still use them interchangeably, but there are some considerations there. Sometimes when we receive a feedstock, sometimes we'll look at it and say, okay, we need to use a little bit more of this, I think. And then that moisture balance, we talked about how our thermophilic stage crashed a little bit in that windrow we talked about. And really our targets are about 40 to 60% moisture in our windrows. So we'd like to stay at 50 or above most of the time. When we add water, we're probably getting up to 60 plus. So it is a range, but that water evaporates and we get transpiration and utilization by the microorganisms too. So we track moisture just as closely as we track temperature and time. And you can use various probes for this. They're going to kind of give you different readouts from one probe to another. I use Dr. Elaine Ingham's method still on this, which is just a simple squeeze test. And maybe I should have put a diagram in here, but I'll try to elucidate what that is. And when we take a handful of compost from near the center, and watch out, it's hot. So if it's during the thermophilic stage, you know, you can burn your hand getting in there too far. But we'll take a handful and we'll squeeze it. And basically for 50% moisture, we're looking for about 1 drop. If I'm squeezing it as hard as I can, I want to see about 1 drop of moisture come out of that. If it's streaming out of my hand, we know we've got plenty of moisture, hopefully not too much moisture. And if we're not getting anything out of it at all, we're either going to, depending on when we're going to turn next, we're either going to top water it. So we're just basically adding water from the top, or we're going to add a good amount of water on the next turn. And I showed a picture of how we're able to add water as we're turning and add moisture to our pile. So we manage moisture very carefully. That's important. You saw what happened when we got a little too dry during the thermophilic stage. We kind of lost temperature. It stalled out a little bit. So monitor that moisture closely in your compost. And then oxygen as a limiting factor. There's just so much activity going on, so much oxygen utilization going on, that a lot of times home composters and commercial operations are just allowing too much static time during that thermophilic stage. And they need to just be dissipating that CO2, reintroducing that oxygen a little bit more often. So leverage thermophilic, then minimize disturbance. We've talked about that multiple times now. And so these cool down protocols, we're still developing them. The one we're utilizing right now is we have an auger that goes on the back of a tractor, and every few feet, we'll just drill about a 10-inch diameter hole right down through the top of that windrow and along that top line of the windrow. And that just allows us to, while we're still kind of hot, there's a lot of activity. We are getting good gas exchange because of these chimneys or aeration ports that we're putting into our compost. But something that we've been playing with more is adding biochar. And right at this stage, and frankly, you know, we can talk about this in a little bit, but if you're charging biochar, if you're if you're trying to charge biochar with compost, or you're adding biochar to compost, like when is the best time to do that? And it seems like maybe the jury's still out on this. I'm sure people have stronger opinions around this than I do, but it seems to me like it's when the compost is still hot. So either you can put it in from the beginning, or at some juncture, sometime, probably mesophilic 2 is what we've been playing around with, and as part of a cool down protocol. And why it has that effect is because it's very electrostatic. Again, the heat and the activity is a function of the food that's left in that compost, right? So we do want to look at the level of decomposition and not stall out that activity too early. But putting biochar in there that's not pre-charged is going to grab a lot of the ions and a lot of the nutrition in that compost. And that is going to greatly decrease the access to that nutrition, and therefore decrease the heat as well, that activity. All right, so that's something we're playing around with. My partner is the one who's really making the biochar at SoilWright at the compost company. But we've added it to some windrows, but we're not adding it to compost presently. So we're also trying to figure out the right percentage. Right now, we think it's probably somewhere around 2 to 5%. But again, the jury's still out for us, and we may change our mind on that. And I don't know if anybody here has any insight into that, but again, if not now, then we can talk about it in a little bit if we have time. So the next thing is, during our last turn, which is typically mesophilic 2, we're adding compost extract. And I don't know how many people here have heard of compost extract. It's similar to compost tea, but you're not actually brewing it overnight and trying to get organisms to reproduce. You're essentially making it by agitating compost particles and knocking the active and viable microorganisms that are already there into solution and immediately getting it into soil. So if you have really good compost and you have enough of it, there's really not a reason to make compost tea. When we make compost tea is a powerful thing. We don't make it and put it in our compost. We basically just make compost extract from compost we already have with a high diversity biomass and population of beneficial microbes, and we inoculate our curing, let's say, compost with that. So we spike in these beneficial microorganisms during that curing stage, if that makes sense to people. Our windrows seem to do fine. We still achieve good microbial outcomes without doing this, but it's just something that we're doing now to ensure quality assurance, because as the last item there says, our quality control is nothing leaves our site until we meet our minimum microbial targets. So we're trying to do all we can for that. That is part of our quality assurance. And right now we're able to achieve microbial levels that are much higher than our minimum targets. But again, that's our guarantee. We're not selling compost unless it's meeting these minimal microbial targets. So again, compost extract to inoculate that during the maturation stage, just to make sure those organisms are there, and we'll continue to proliferate on that lignin and those food sources that are still left in the windrow. And then here's one that probably I don't think too many commercial operations would be very fond of this, because real estate on a compost pad is high. And so they want to make compost, and then they want to get it off the pad and over into a big pile in the corner. And that's sort of their saleable compost at that point in time. But what we do is we actually have enough room to store in windrows. So we don't put it in a pile or move it once it's mature, once it's ambient. We allow the fungi really, but all those organisms, to continue to do the work on the lignin that is undoubtedly still there. And then we screen it just before we sell it. So as we pull in orders, or as we know what we're going to do for that week, we do screen it down to just under an inch. but we leave it stored in a windrow with those large chunks of wood chips in there for continued fungal proliferation until it's sold. And so one of the pitfalls that I've seen with moving it off to the side and storing it in some of the, I don't know if anyone here has seen like these big mountains of compost. Francesco, did you have a question? You can go ahead and unmute yourself and ask it.
Yeah.
Thank you.
So how do you know you met your minimal microbial targets? Do you send it out for testing?
We actually do the testing in-house, and then we do third party as well.
Okay.
So for our quality assurance, we are quantifying what's going on with our compost very closely and very often. It is in flux, it changes. We know that our customer wants to see third party results though. So we do third party on that. And then, so that's what I have the best feel for. And it kind of is like what you see is what you get. So, I can see that these protozoa are actively there. I can count them out and I can quantify their population. Same with nematodes, same with bacteria, and same with fungi, where we also do DNA testing. And the reason we do that is because we want to know the species that are there in our compost. We most of the time cannot tell down to genus or species level what these organisms are that we're working with, unless like some of these nematodes, some of the protozoa, and some of the fungal species that we're very used to seeing, we can do that. But we are doing 16S and ITS, genomic or PCR next gen sequencing, and they're able to tell us what these species are that we're counting out in our compost. The other reason we're doing that is because through the Oregon Department of Agriculture, we are in the process of getting our compost registered as a microbial inoculant. In the past, they had not been accepting this genomic data that we're collecting, the 16S and ITS, where we get these bacterial and fungal species. But now they're sort of opening that door a little bit, which is great because in the past, they were only allowing for cell culture. And so basically culturing them on media and then counting out the colony forming units via microscopy is what most agricultural departments are still doing, and not really looking at genomics or PCR for verification of, whatever you're saying in your guaranteed analysis on your label. So they're just kind of opening that up. We were already collecting that data, but now we're collecting it more frequently because we want to list more of these species on our label, and it looks like they are going to allow us to start doing that. Now, there's seasonality in the microbial makeup and the species and the level and everything, so we have to do this regularly and adjust our label, probably at least twice a year, once or twice a year. So that's what that process looks like for us, but that's another way we're verifying what's going on in our compost. Yes, Mary, question.
Yes, thank you. So what is the size of your windrows and what do they look like?
Yeah, that's a good question. I should have put a picture of those in here. Well, I'll sort of try to draw a mental picture for you. And you can go to I'll show our website. It's SoilWright.com. You can go and see some of our windrows. I think they're undercover mostly in the photos. We're a little media light and we've just made our website. So we need to get some more photo assets and add them to the website. But so I'd say 4 to 5 feet tall. So peaked, right? So like a long triangle, if you will, and probably 6-7 feet wide, sometimes 8 feet in the beginning. They're bigger in the beginning before decomposition happens. And then we end up losing about 40% of that biomass and size, those dimensions, by the end of it. So it's very condensed, but we also lose a lot of that carbon, unfortunately, to the atmosphere. But yeah, so about 4 or 5 feet tall, 6-7 feet wide, and up to, I don't know, let's say 120 feet long. And we'll probably make some longer windrows than that as well. But yeah, if we'll show the website address here in a minute, and you can look at some
Is
there.
there a reason you chose windrows over piles?
Efficiency, space utilization, yeah, and just when I, it was a little bit of a learning curve to go from piles to windrows a few years back, but I realized that I can almost do better in a windrow than I can in a pile. And I've turned a lot of compost by hand over the last 10 years. Um, I could not turn compost by hand in this case. I don't prefer to turn with a tractor bucket mechanically because you just can't, our windrow turner is actually making like a hop pattern. So it's taking the bottom and the middle and it's throwing it up and then it's churning the outsides into the middle. So after about 3 turns, everything has made it through that hot center. So it's very efficient at doing that and creating just a more, I don't know about homogeneous, but a consistent product where everything's making it through the middle, and we're still getting these great microbial outcomes. And again, we're mid-scale. I don't know where we go from here. I guess we'll cross that bridge when we come to it. But yeah, those are the main reasons we've selected for mechanization. We make presently a lot less compost than most large scale or municipal composters because it does cost more, because of this focus and this outcome. But at the same time, a little goes a long way when you're making it as an extract. For example, if you were treating seed, you could maybe use a couple ounces of extract per acre to treat that seed coat. Go out on broad acreage with a seed drill, and put that out on hundreds, if not thousands of acres we've seen in some cases. And sometimes we'll make an extract which can go in furrow. So basically putting it right on the seed as the seed drill plants it out in the field. We've used as little as about 40 gallons of extract per acre, multiple times per year, and usually in conjunction with other foods or biostimulants or organic or mineral inputs that are in nutrition program for that crop. But a little goes a long way. We really are trying to make inoculation grade compost, and it goes very well with other types of bulk compost. So compost that maybe doesn't have as good of a microbial outcome, but it still has carbon, organic matter, nutrients in it, still good for your garden. And then you can come through and sort of add the life with a compost more like this. So that's really what our product is.
So Nick, does that make it so that the soil stays alive better than a lot of commercial outfits? Because a lot of their systems are, because they're so anaerobic and they're so hot, they kill everything. And I mean,
Are
so
you, yeah, are you talking about commercial composters?
yeah.
Yeah. And again, that's why we have to manage that thermophilic stage so carefully, and really introduce that oxygen, and not put too much food in there, not get too much activity, make sure that carbon's there so we get that proliferation of those other organisms on the back end. But I mean, if you think about it, if you've sort of grasped the way we're composting and a little bit about what some of these other composters are doing on even a larger scale, it's relatively simple adjustments to achieve a better outcome. It's just the will first of all, the intention and the goal has to be there. You have to know why you're doing those things. But I've worked with a number of different commercial composting operations and have had varying levels of success depending on what kinds of changes they're willing to make. And not everything we're talking about here that we do would have to be done, but they do need to manage that recipe a little differently, get some more carbon in there, and manage that thermophilic stage with the reintroduction of oxygen a little more closely. But yeah, I don't think it would take too much to bring some of these bigger municipal composters into producing a product with a better microbial outcome.
Yeah, it's really thinking about compost to soil health instead of, just a way to reuse waste.
Yeah.
Yeah.
And I tried to work in some like
Understanding that ecology
ecological.
is,
Oh, sorry, Catherine, go ahead.
yeah, understanding that ecology is huge.
Yeah.
And I think the fungal piece is so new that it's
Yeah.
really not entered that conversation very much.
Yeah, I think when we're talking about commercial composters and then like home composters in a lot of cases, yes, you're right. You're absolutely right. For people that have been making compost, for people that are into permaculture, regenerative, I mean, that is a buzzy aspect of the conversation around compost for sure. And it seems to me where there was like some sort of adversaries in this regard within the regen space even. It seems to me that it's just proliferating or myceliating through that community, if you will, more and more as time goes by. And so it's been great to see that for sure. Okay, so biochar. This is a picture of one of the biochar piles we kind of still have sitting, and we do have a hammer mill, and we'll break this down into, we're just getting it operational now, but we'll break this down into some smaller particles as well as we decide how to best utilize it. If we want to put it in all of our compost at 2%, if we want to offer like a different compost product, the hat plus biochar or something like that. But yeah, with biochar, as I sort of alluded to before, there's many different feedstocks that can be used to create biochar. I mean, I've seen manure, I've seen wheat straw, I've seen chaff, I've seen wood chips, obviously different types of wood. There's many methods for, well, pyrolysis is pyrolysis, but I see a lot of, we have a DIY reactor. We don't have some expensive bioreactor right now to make this, or biochar reactor to make this. They're pretty spendy. And I'm not sure we'll get there. It's definitely a topic of conversation that has been revolving for a while for us. But with regard to like the methods of charging it, I have tried to go slow with biochar as I try to get a feel for it, and I've sort of been pulled into it from all sides, whether it's from clients, peers, partners within SoilWright, for example. And yeah, so but there's a few projects right now where I've consulted on the charging of biochar, and in one case there's a vineyard that has just been planted this spring, and we charged it up. We did a bunch of things with the soil and cover crops, etc., but we charged this biochar and some compost extract. We added some nutrition. We tried to get the C to N ratio where I thought it would be somewhat ideal, and we put some minerals on it to kind of hold on to that, and so we'll see how this winter does. But I've had go ahead. Oh, sorry. Go ahead, Francesco. You have a question?
Yeah, I do. What is you made that biochar in this picture?
This is my partner made this biochar. Yes.
Okay. What was his feedstock, and what did he use when you say pyrolysis? Was it a flame cap kiln, or was it a top lit updraft stove, or what did you use?
It's sort of his own design, like many things with our operation. He's got this engineering mind, and so it's only semi-enclosed. So I'm not sure that it's actually an oxygen negative type environment like a lot of these reactors are. Okay, so this probably borders on charcoal biochar, I would say. But let's see, he used kiln dried wood to make this biochar, and it's basically pine and fir.
So Kelsey Wilson is somebody I would recommend, and she has a book and she has a company. She's in Washington, and she has created the Ring of Fire kiln, and it is open. It is okay to use in the state of Washington. They changed the rules just for her kiln, actually. Because the way it's designed, it keeps everything in. So even though it's open, it doesn't emit sparks. It doesn't emit very much gas at all. It keeps everything in. So it's the way it's designed to do that.
Yeah, that's the way he's tried to design his as well. And you've mentioned her before, and Kelsey Wilson, is that what you said?
Kelpie, Kelpie Wilson.
Kelpie Wilson, gotcha.
And her, the Ring of Fire is around $2,000, I think. So it's really affordable. It's movable. You can put it in the back of a truck, take it apart and put it, because it was designed for forest use. They can easily cart it around. So I highly recommend her. She's been in several of our webinars.
Okay, and I would just like to. I've worked with biochar for over a decade now, and when you say how I tell people the simplest way to charge biochar is let nature do the work and co-compost it. Put it in your compost as you're starting your compost pile. When the compost is ready, the biochar is charged. And you can put in somewhere, and you mentioned somewhere between 2 to 5%, because as you mentioned, Nick, the pile is going to diminish. So if you say 50%, if you put in 5%, you put in 5% biochar, you're going to end up with about 10% biochar by volume in your finished product.
Yeah, right. Because everything else decreases, but not the biochar.
Exactly. And so, and there are people, are you familiar with Rexius commercial?
I am.
Okay, well, they did some stuff years ago, did some tests, some trials, co-composting with biochar in static piles, and they found that they had a more active compost in the pile that had about 5% biochar versus their control pile. They had a higher microbial, bacterial, and fungi life in the compost with the biochar. So
Yeah. Thank you. Thank you for that information.
Charles, do you have a question?
Yeah, go ahead, Charles.
Yeah, I do. So I guess just at a very basic level, is there a good way to test biochar? I guess I have a lot of clients and they say, "Oh, I'm making biochar," and I'm like, "Well, I don't think you're really doing pyrolysis there. That's more of a charcoal." Is there a good way to like just take a look? Is that a microscope thing, or do you have any ideas there? I'm just like questionable. I think it's kind of gotten out into the,
Yeah.
People burning,
think-
thinking it's biochar, and also not charging too. So.
Right. I will say one comment. I will say, my partner Robert, who made this, he's pretty adamant that it is biochar, and I believe him. He spent a lot of time trying to figure out what he was doing here. He's still not all the way happy with it, and it may be a little on that charcoaly spectrum, but he's pretty proficient. He's got a lot of time into it. But I would say that probably Francisco might have an answer to your question there. I know one lab that can do the testing, but it's like the Biochar Institute. You guys are going to know more about this than me.
if you've done it it sounds like your partner did it in some sort of a type of flame cap kiln, you are going to make a biochar as long as you're achieving temperatures of at least 400 degrees. And then one of the issues with homemade biochar is that the quality is completely dependent upon the feedstock, the moisture content, and the operator that's doing it. And there are some commercial biochars, and I'm fortunate enough for years was getting and still do, but I sold the business, getting the product of the biochar from a paper mill here in Port Townsend. And it's a byproduct of a steam boiler. It's a high temperature biochar and there's different testing labs that you can send them out to. Soil Control Labs is one of them.
That's exactly the one. Yeah, that's where we test for our compost testing, the bioassay, the respiration, the mineral nutrient, all that stuff. We do Soil Control Lab, and we're going to send our biochar in there soon. But we just have yet to do it. And again, we're going really slow. We're not adding this to our product until we know what's going on with this.
You don't have to add biochar every year either. I mean, it's not something that you have to put on every time you dress your soil. So I would want compost without biochar as well.
I agree. And I'll share one quick story. So I don't know how many people here have worked with a micronized biochar or like a solution grade biochar that you can put into liquid and water in. We were working on a project where we used a little bit of this, and I used quite a bit of, what did we do? So we tank mixed it. We put in quite a bit of soy protein hydrolysate. We put some kelp and some minerals and things in there. We had some data that we were working off of, some sap data and some soil data. So we we're trying to sort of formulate this thing around that. And I decided to add even a little bit more nitrogen. I'm usually pretty light on nitrogen, just like with composting in my nutrition programs for crops. I'm trying to utilize less nitrogen, and especially over time. But anyway, what we saw with this, and it was in an engineered media, so low bulk density media, which was part of the issue, was it turned that we were able to turn around from this, but it turned that crop sort of yellow in a matter of about 48 hours because it just grabbed onto so much of that nutrition that had been flowing in through mass flow. And so it was a big lesson for me. Like I said, we were able to turn around quickly on that, but where here I thought this was one of my earlier experiences with trying to charge it and utilize it, but in a liquid form. And right then I knew the power of this stuff and that it needed to be respected. I needed more experience with it. And I needed to really know what I was doing with biochar before I was recommending it, charging it, et cetera. So again, hence my comment that I've tried to go really slow with biochar and gain experience.
Well, and as I
I
say,
want to give a little shout out here. For June 25th, we will have Dan Donnelly. And Dan has spent 30 years using biochar on golf courses. So if you know anything about golf courses, they're really picky. You have to be really careful about that grass. And so he has learned a lot about how to apply it and do that. So he might have some answers to this kind of stuff. So just a shout out for that.
But just, so when I met Catherine, she reached out and I don't remember like the reason you first reached out, but I know we quickly were talking about a project that you were working on, and there were some contaminants. It was in an urban area, and we were like, talking about remediation, bioremediation, micromediation, even a little phytoremediation. And I think for me, like the biggest problem with that one right up front was that we didn't know what the contaminant was. So that was part of that would be part of the project, would be to identify and quantify what actually the contamination or contaminants were. But we started talking about it and looking at biochar and trying to figure out, how this would work. And I think Catherine, you're still working on this for a solution for this, perhaps, but we haven't talked about it in maybe a month and a half.
Well, and when you get into remediation with biochar, then you have to figure out what to do with what you've taken out of the soil.
Right. And that's kind of where we were stumped. Right?
Yeah, and that kind of stuff. So yeah, so it was a little above our pay grade at this point in time. So it's a big deal we still nurture but haven't grasped yet.
No.
Yeah, and I personally have not used biochar in the soil, but I've used biochar to remove heavy metals from stormwater runoff.
Very cool. And I was gonna, continue, elaborate.
And we just made downspout filters at the Port of Port Townsend, and we were removing zinc and copper.
Yeah.
What did you do with the biochar after?
Yes.
It's still there. They've abandoned, but the Port of Friday Harbor is doing the same exact thing with systems that they've designed. And with the biochar that has reached breakthrough, they just take it and they've got a wooded area and they spread it in a wooded area, because the biochar, the spent biochar can be disposed of in a I think it's called subtitle D landfill, which is construction waste, non-hazardous. It's not considered a hazardous waste because it
would hang on to the metals.
Excuse me?
It would hang on to the metals.
Exactly. It hangs on to the metals.
Yeah. But yeah, so I just, that is a question for me. I really want to feel that there's this full cycle somehow or other, that we're not just collecting piles of waste someplace, but there is some way to begin to release that back into nature in a way that's not harmful, but is beneficial. And I don't know that we know how to do that yet. So I'm kind of really cautious around that. But I also want to make a little segue into one of the wonderful things about Nick, and one of the reasons I reached out to Nick, and one of the programs that we have here is because Nick's training at, and specifically the Soil Foodweb school, is such a rich and deep understanding of soil and soil health and the life in soil and how to maintain that and to have it thrive. And I haven't seen that in any other place. And I think I'm hoping one of the things that people have gotten out of tonight is that compost is so much more. It has so much potential to really make a very quick difference in our very, very, very depleted soils. So much of the soil around us is just hanging on by a thread. And it needs so much more than what we have really understood it needs. And I think this is one of the things that comes from Nick's talk, and it is so inherent in how the Food Web School teaches. We are offering scholarships for 2 people to go to that school who live in Spokane area because we need these resources here. Nick's in Oregon, and he's been just wonderful and is very helpful. But it would be really wonderful if we had people here. So I'm encouraging people to think about a career in soil science. A lot of this is microscopic. It is learning how to see those things in the soil so that you actually know what's there. It's not about guesswork. It's about being able to actually know for sure through data what you have. And that's a step above what we've been able to do in so many ways. So it is really great to be able to see and such a great example, and how far you've taken that, Nick. Yeah.
Yeah, it's been a long road, and I met Elaine. I went to Oregon State. She was a professor at Oregon State, so I was able to buy compost from Dr. Elaine when I was an undergrad, and I was using it in gardening projects and various. And I was just like, that's when it was kind of an aha or eureka. I was like, this is a real thing, and then I went like 10 years thinking that I could just do these things on my own. I was in agriculture the whole time. I was doing various things around, agronomy, mineral balancing, crop nutrition programs, crop plans, that kind of thing. And just kind of assuming that I was proficient on the biological side because, of things that were anyway, until I really circled back to that. I didn't really have those pieces in place the way I thought. And now that it's been 10 years since I had trained, there were only, I don't know, maybe a dozen people or something back when I did that training, and then they created this new school after, and now there's hundreds of people, if not thousands of people around the world who are doing that, which I think is amazing. But yeah, it just came at the right time for me. For sure, when I was really trying to work on those pieces, but I was struggling to put them together with what I could find in the literature, what was going on within research, conversations with peers, and even scientists in some cases. So it really was a turning point for sure.
Which is pretty amazing. Charles?
Yeah, I'm wondering if you have any opinions about the microbiometer, if you're familiar with that, and for soil testing. I've been using it, and it's kind of fast and easy, but is it accurate, I guess? I have some microscope skills, but they're kind of lacking. I could improve there.
I have limited experience. I think everything has its faults for sure. There are questions that remain with any type of microbial testing that I've done, and so PLFA, phospholipid fatty acid test, that's another one we'll pull sometimes for some projects. We do that genomic, which I think is good, but it's expensive. I mean, you can get up into $300 to $500 for tests, once you go to that side of things. The microbiometer, I've only used a few times, and so I feel like it would be unfair for me to try to give an opinion on the microbiometer. I didn't see in those few instances that it was correlating neatly with what I was finding via microscopy. But I didn't have PLFA data to compare those particular instances to, which I think maybe would have been good. So depending on where your projects are, I do my PLFA testing through Regen Ag Labs in Nebraska. That's also, I've been doing more testing with them. They do Haney testing there too. So sometimes I'll pull both of those from them. And then Melic 3 is what I normally use for soil. So, and they do Melic 3. So if I'm already using them for PFA or Haney, I'll just have them do the Melic 3s too. But they've been a good lab to work with, and PLFA might be something to look into to at least correlate what you're finding with the microbiometer.
I mean, for my use, I mean, I can do it on site, you know, very quickly,
Right.
right? And I don't have to send
Right.
it out. So I'm usually, so it just gives me that kind of snapshot, but I'm always, I get the question as well, like, how accurate is this? You know, and they claim
Yeah.
90 something percent. Anyway, I'm just curious if you had opinions about it because I
Yeah.
get that question. I like the test. It's only, I think it works out to be like $7 a test or something like that, maybe even less. So it's
Yeah.
fairly cheap and fast, right? So that's the
Yeah.
big benefits.
I could see the benefit being tracking data over time, just like with anything, you know.
Mhm.
What's your baseline data? What are you doing? What are your goals? You know, what have you done to try to achieve those goals? And then are you seeing that you're moving closer to that goal with microbiometer data? Um, so that's, yeah, I was really only suggesting the PLFA too, just as maybe like a proof on the microbiometer, just to see if there's, do a couple of tests, run some correlation there, see if you're kind of close.
Sure,
Again, at the same time, it could take some pretty extensive testing to get a feel for the two and how closely correlated they are.
sure. Yeah, thank you. Appreciate that.
Yeah.
Thank you, Nick, so much. So I want to give a shout out to the webinars we have coming up. Please check out the website. Every 2 weeks on Wednesday at 5:30, we're doing a webinar. The next one will be Michael Polarski, who's talking about the food food forests and the bionic pump for those of you who are into permaculture. And then we'll be having Charles actually, will on June 11th, we'll be talking about how to keep the water on the land, which is a really big piece of it. So it's our human beaver approach to things. And then on the 25th, we'll have June 25th is Dan Dinelli talking about biochar on golf courses. You can support us and support the work we do in a number of ways. You can become a member. If you join the community, which is what's holding these webinars, it's $10 a month. That's really helpful. You can also donate to either of these three projects. So the center one is us, but if you want to add to our forest planting pool, then donate to Plant Forests. And if you want to support people in careers in soil science and water hydrology, we have one scholarship there as well. You can contribute to that also. Those of you who are here, if you keep your URL, you may come back and watch this at any time. But it is only through that URL that you can do it. If you become a member of the network, then you can access this and all of the other free webinars at any time that you wish. So just a little FYI, you want to keep that if you want to come back and view that. Nick, thank you so much for your time. We really appreciate it. And
Absolutely. It's a
yeah, it's
pleasure.
amazing to hear the things that you've done. And I just really hope people appreciate the depth that you have come with in terms of understanding the life in compost and how to keep it there.
Yeah, the truth is I'm probably still early in all this learning, and there's much, much, much more to learn. So something to look forward to for sure. But thank you so much for having me. I really enjoyed it.
Thank you. And thank you, everyone. Oh, Carl.
Wow, that was intense.
Yeah, he's pretty amazing, isn't he?
Hmm.